visible?
Yes.
Hello, my name is Ritu, and my topic is Taylor Woods and Hughes experiment. In the year 1958, Messrs and Stars performed an experiment using heavy isotopes of nitrogen. With that, they concluded that DNA replicates in semi-conservative form. Later, a similar experiment involving the radioactive thymidine to detect the distribution of newly synthesized DNA in the chromosome was performed on the root tip of vesica faba by Taylor and its colleagues. The experiment also concluded with the same thing that DNA in the chromosome replicates semi-conservatively. In the experiment the unlabeled chromosome was replicated in the radioactive thymidine in the first cycle, where we got the first chromosome with the chromosome with radioactive thymidine label. In the second cell cycle, it was again replicated, but in the absence of radioactive thymidine, where we got that the half cysteine chromatin was labeled with radioactive thymidine, and second half was normal thymidine present, which uh, which concluded at the replication is in semi-conservative manner, where one side is parental and the second half is daughter cell. So that's it. So hydride. Uh, yes, sir. Huh. So cesium क्लोराइड यूज करके ये किया था. Nitrogen chemical form. No, I guess so. हाँ. सर जो वेव्स प्रोड्यूस होते हैं रेडियो वेव्स प्रोड्यूस होते हैं उससे वो क्या बोलते हैं. हेलो.
Yes, sir. Yes, sir. Okay, sir. One sec. Sir, is it visible? Yes. Yeah, just. Hello everyone. My topic is enzymology of DNA replication, and I'm going to talk about three enzymes: topoisomerases, SSB proteins, and TUS proteins. So first, I'll give some information about DNA replication. DNA is a self-replicating molecule, that is, it has the ability to replicate by itself. DNA replication is the process by which the genome's DNA is copied in other cells. Our DNA replicates semi-conservatively, that is 50% of our DNA is of the parental generation and 50% is new DNA. So the replication of DNA is very important for the transmission of genes from the parent DNA to daughter DNA. Enzymes play a very important role in DNA replication. So the enzymes I'll be talking about are topoisomerases, SSB proteins, and TUS proteins. So in topoisomerases, what happens is that the activity of the helicase leads to supercoiling of the DNA ahead of the replication fork. So to release the tension that is created because of the supercoiling, topoisomerases are activated. So the amount and type of DNA supercoiling is controlled by topoisomerases. These enzymes are found in all organisms. They relax the tension in the DNA ahead of the replication fork. They cut the DNA strand, relieving the tension, and then again they have the ability to reseal it. So there are basically two types of topoisomerases: type 1 topoisomerase and type 2 topoisomerase. So in type 1, what happens is that type 1 can cut only one strand. Then the 5-phosphate end of the strand is attached to the tyrosine molecule in the topoisomerase protein, and the 3' end passes to the duplex once, and then it is resealed. So the tension is relieved. In E. coli, type 1 can only relieve negative supercoiling, but in eukaryotes, type 1 can relieve negative as well as positive supercoiling. So type 2 topoisomerase, they can cleave the DNA duplex, that is, they can cleave both the DNA strands. They have the ability to convert positive supercoiling into negative supercoiling. These topoisomerases are also involved in DNA in decatenation of the DNA molecule. DNA gyrase is an example of type 2 topoisomerase. So in this diagram, we can see that topoisomerase can cut only one DNA strand and two can cut both the DNA strand. Topoisomerase 4 is involved in the decatenation of the DNA. So now SSB proteins, single strand binding proteins are the proteins which bind to the unwound single stranded arms of the DNA of the replication fork. What happens is that when helicase unwinds the DNA molecule, there is possibility of reannealing of the two single-stranded DNA. So what happens is SSB proteins bind to the single-stranded DNA and prevents them from reannealing. Small SSB is the gene which codes for these proteins. Then here we can see this purple color small molecules are the SSB proteins, which are bound to the single-stranded DNA and which prevent the reannealing. So now TUS proteins. TUS protein is a protein which binds to the TER sequences or the terminator sequences, which act as a counter helicase. Counter helicase because helicase is a molecule which starts the replication, and this terminator proteins or terminator sequences this halt the replication fork, stopping the replication. The TUS proteins have the ability of halting the activity of the DNA polymerase. TUS proteins help to end the DNA replication in prokaryotes. Small TUS is the genes which codes for these proteins. In E. coli, TUS protein binds to the 10 closely related sites encoded in the chromosome. These sites are known as TER sites. So they form a complex known as the TUS-TER complex. This complex encounters the replication fork and halts it. So multiple sites are present in the chromosome, and they are oriented in the opposite direction to stop the replication, which is advancing from both the directions. So in this image, we can see that this Terster complex is present. The non-permissive phase of this complex has the ability to stop the replication fork. And sometimes what happens that the Terster complex is not able to stop the replication fork. So the replication fork just moves forward. So therefore, multiple Terster complexes are present on the termination sites of the chromosome. Thank you, sir. Yes, sir. Sir, if there is no termination site, then both the replication forks would overlap and the replication wouldn't end. Terminus utilization substance. Sir, decatenation is basically when two DNA molecules are intertwined into each other. So the topoisomerase basically cuts both the strands and separates both the DNA molecules. So that is decatenation, separation of two DNA. Topoisomerase 4. Thank, thank you, sir. Thank you.
Yes, sir. No, sir.
Yes, sir. My name is Monali More. My topic is failure roots and of experiment. It's the story of person and field along the structure of DNA. It was more the thing that the way theory in the molecule of DNA, but the question now arises how the structure of DNA replicates. Further, with the study of Taylor and his colleagues in the year 1958, the semi-conservative mode of replication was seen. The Taylor and his colleagues in the year 1958 also demonstrated the semi-conservative mode of replication in the DNA and chromosomes in its cell of Lucia faba. After the incorporation of radioactive cyanide, it creates the root its cell transfer to unlabeled medium containing radioactive chromosomes with labeled DNA having a CH appearing in the form of that a black dot of silver grains. After replication of DNA and constitution of chromosome, following facts are noticed.
First, it is the found in first generation that radioactive was uniformly distributed in both the chromosomes. In such a case, original strand of the DNA, a double helix was labeled with CH and newly formed strand was non-labeled. Second, during second division, only one of two chromosomes represented the radioactive by showing one standard radioactive and other one non-radioactive newly formed. This is dictated by the arteriography technique. This implies the ionization of silver, a light sensitive compound on radioactive exposure. The silver atoms and form get the deposit on the X-ray fields. Chromatography is technique used to separate two or more substances in the mixture which have a different degree of solubility in the given solvent. Taylor and Hooks and House showed that DNA replication is semi-conserved in cell of Lucia faubabil and using the technique of hyperradiography. Thank you, sir. Uh, at yes, sir. Arteriography imaging the prescription of the silver atoms resulting from the ionization of silver halide by the radio label sample uh, light sensitive compound commonly used in photography. Uh, electric motor and the
Yes, sir. Sir, it is visible. Okay, sir. Sir, now it is okay. Hello, sir. Yes, sir. Sir, it is visible. Okay. Good evening, sir. Good evening, everyone. I am Miss Akanksha Navnath Nakwa, and my roll number is 227321. And my topic name is Replicons and Initiation of Replication in Eukaryotes. Replicons. So, a unit of DNA replicated by two replication forks from a single origin is termed as replicon. A replicon was conceptualized as a unit of replication that consisted of a DNA sequence called the replicator, now called origin of replication or ORI, and it is also called as an initiator, a protein that interacts with the ORI to initiate the replication bubble. The chromosomes of archaea or eukaryotes have multiple origins of replication, and so their chromosomes may consist of several replicons. For eukaryotic chromosomes, there are multiple replicons per chromosome. Known examples range in size from 10 to 330 kilobases. A cluster of replicons replicate simultaneously. The concept of the replicon was formulated in 1963 by Francois Jacob Sidney Bernier and Jacques Cousin as a part of the replicon model for replication initiation. So next is initiation of replication in eukaryotes. DNA replication initiates from specific sequence origin of replication ORI called replisomes. Eukaryotic cells have many multiple replication sites. To initiate replication process, multiple replicative proteins must assemble on their replication sites. It leads to formation of pre-replication complex, that is pre-RC. So for pre-replication in the replication, as we see in the diagram, uh, there is a DNA molecule. It is a linear in eukaryote having two strands running in an opposite direction. And also in this diagram, you can see a site called as ORI. Uh, ORI site, that is origin of replication. This is where the replication starts. Uh, in this initiation, there are some proteins involved that are ORC protein, CDC6 protein, CDT protein, CDT1 protein, MCM27, CDK and DDK, that is cyclin dependent kinase, and DBF4 dependent kinase, and GINS, that is a co-activator of initiation, and uh, and RFC, that is replication factor C. Uh, Here, pre-replication complex at stage, that is association of origin recognition recognizing complex, that is ORC, with replication origin, binding of CDC6 protein to ORC, then binding of CDT1 and mini chromosome maintenance protein. Then in pre-S phase, uh, there is a phosphorylation of MCM and MCM protein and CDK6 protein, and this replicative complex assembles occurs during G1 phase prior to S phase. During the transition between of G1 phase to S phase, CDK proteins and DDK proteins get attached to the pre-replication complex. It transforms the pre-replication complex into active replication fork, and there is in S phase, uh, in S phase there is a bubble which, sir, thank you sir. हाँ sir. Yes. Uh, Full form of CDK is cyclin dependent kinase protein. Sir, it regulates the initiation protein by phosphorylation. Phosphorylate to, sir, MC, हाँ sir, MCM27. हाँ sir, cyclin. सर ARS भी रहता है. हाँ सर, ORC रहता है. ओरिजिन. सॉरी सर. Sir, DNA replicates in. Sir, you asked about replicate and separate, right? It replicates in uh, elongation phase. So, हाँ हाँ. DNA sir in S phase. Uh, S phase and सर और एक होता है. And sir separates in. It's, uh,
Yes, sir.
कौन से वाले सर? याद आ सर.
Yes, sir. Okay, sir. Wait a second. Good evening, everyone. Myself Mayuri, and today's my topic is rolling circle replication. So, in rolling circle replication, some viruses chromosomes, such as that of bacteriophage, a circular double-stranded DNA, replicate to produce linear DNA. The process is called rolling circle replication. So, you can see in a picture how rolling circle replication rapidly synthesize multiple copies of circular molecule of DNA and RNA. So it is a process of unidirectional nucleic acid replication that can rapidly synthesize multiple copies of circular molecule of DNA or RNA, such as plasmid, the genome of bacteriophage, and the circular RNA genome of viroids. Some eukaryotic viruses also replicate their DNA or RNA via the rolling circle mechanism that are bidirectional. So here is the steps of rolling circle application. In first step, you can see in a picture, the rolling circle application is a generation of specific nick or cut in one of the two strands at the origin of replication. In second step, the 5' end of the nick strand is then displaced on the circular molecule to create a replication fork. In the third step, the free 3' end of nick strand acts as a primer for DNA polymerase to synthesize new DNA using a single-stranded segment of circular DNA as the template. The displaced single strand of DNA roll out as a free term of increasing length as the replication process. New DNA is synthesized by DNA polymerase on the display. In the fourth step, DNA is 5' to 3' direction, meaning from the circular out toward the 5' end of the displaced DNA. With further displacement, new DNA is synthesized again, beginning at the circle, the moving outward along the displaced DNA strand. Thus, the synthesis of the strand is discontinued because the displaced strand is lagging strand, template as a single standard DNA tongue rollout. New DNA synthesis proceeds continuously on a circular DNA template. Because the parental DNA can continue to roll, a linear double-stranded DNA molecule can be produced that is longer than the circumference of the circle. So here is the overall explanation of rolling circle application. In the first step, see the how DNA will leak. In the second step, 3' end elongated. In the third step, 5' end displaced. And in the fourth step, replication DNA circulates and synthesis of its own complementary strands. So here is the basic steps of rolling circle application with initiation, elongation, and so in initiation, phosphatase end by the action of helicase topoisomerase single-stranded binding proteins. In a second elongation, OH group of broken strand using the unbroken strand as a template. The polymerase will start to move in a circle for elongation, due to which it is named as a rolling circle model. So end will be displaced and will grow out like a waving plate. Third is the termination. At the point of termination, the linear DNA molecule is cleaved from the circle, resulting in a double-stranded circular DNA molecule and a single-stranded DNA molecule. The linear single-stranded molecule is circularized by the action of ligase and then replication to double-stranded circular plasmid molecule. So let's consider the example of Phage lambda. So Phage lambda has a linear, mostly double-stranded DNA chromosome with 12 nucleotide long single-stranded ends. The two ends have complementary sequence. They are referred to as a sticky end because they can pair with one another. When Phage infects E. coli, the linear chromosome is injected into the cell and the complementary end pair to produce copies of chromosome to pack in a progeny phase. The now circular phage chromosome replicated by the rolling circle mechanism. Thank you. Yes, sir. E. coli. So both कंडीशन में रेप्लिकेट होता है. bacteriophage, uh, 5x174, single stranded RNA है ना सर में. सर एक बार और बताइए. नहीं पता सर. सॉरी सर, इकोलाई फैक्टर एफ ना.
Yes, sir. Yes, sir. Okay, sir. Sir, is it visible? Hello, sir. Okay, sir. Myself, Khushali Parke. My roll number is 227322. My topic is eukaryotic replication enzymes. Here are the list of enzymes in eukaryotic DNA replication. First is helicase that unwinds the DNA helix at the start of replication. Second, SSB proteins, which binds to the single strands of unwound DNA to prevent reformation of the DNA helix during replication. Next, DNA polymerases. Eukaryotic cells contain 5 different DNA polymerases: alpha, beta, gamma, delta, and epsilon, which are in detailed in further. Telomerase, a DNA polymerase that contains an integral RNA that acts as its own primer, is used to replicate DNA at the ends of chromosomes. DNA topoisomerase 1 relaxes the DNA helix during replication through creation of a nick in one of the DNA strands. DNA topoisomerase 2 relieves the strain on the DNA helix during replication by forming supercoils in the helix through the creation of nicks in both strands of DNA. Last is DNA ligase forms a 3' to 5' phosphodiester bond between the adjacent fragments of DNA. At the eukaryotic replication fork, there are 3 distinct replicative polymerase complexes that contribute to DNA replication. They are polymerase alpha, delta, and epsilon. These 3 polymerases are essential for viability of the cell. Replication of each linear DNA molecule in the chromosome starts at many origins, one every 3 to 300 kilobase of DNA, depending on the species and tissue, and proceeds bidirectionally from each origin. At each origin, a replication bubble forms consisting of 2 replication forks moving in opposite directions. The DNA replicated under the control of single origin is called replicon. DNA synthesis proceeds until replication bubbles merge together. At the origin, enzymes unwind the double helix, making its components accessible for replication. DNA polymerase alpha, also known as Pol alpha, is an enzyme complex found in eukaryotes that is involved in initiation at origins of replication and during synthesis of Okazaki fragments on the lagging strand. Pol alpha complex, that is a Pol alpha DNA primase complex, consists of 4 subunits. The catalytic subunit Pol A1 the regulated subunit Pol A2, and the small and the large primase subunits prime 1 and prime 2 respectively. Once primase has created the RNA primer, Pol alpha starts replication, elongating the primer with 20 nucleotides. Here is the diagram which shows the elongation. DNA polymerase beta. In eukaryotic cells, DNA polymerase beta performs base excision repair required for DNA maintenance, replication, recombination, and drug resistance. The mitochondrial DNA of mammalian cells is constantly under attack from oxygen radicals released during ATP production. Mammalian cell mitochondria contain an efficient base excision repair system employing Pol-B that removes some frequent oxidative DNA damages. DNA polymerase delta is an enzyme complex found in eukaryotes that is involved in DNA replication and repair. The DNA polymerase delta complex consists of four subunits, POLD1, POLD2, POLD3, and POLD4. DNA pol delta is an enzyme used for both leading and lagging strand synthesis. Besides its essential function on the lagging strand of the DNA replication fork, this enzyme also functions during DNA recombinational processes, various DNA repair processes, and even during damage, including mutagenesis in the cell. POLD1 is a catalytic subunit with polymerase and exonuclease functions and is critical in several synthetic and DNA repair processes. POLD2 with POLD3 and POLD4 regulate the activity of the complex by stabilizing the structure, establishing protein-protein interactions, and regulating the cell cycle. DNA polymerase epsilon polymerase composed of four subunits synthesizes the forward strand during conventional DNA synthesis. DNA polymerase epsilon fulfills roles in the DNA synthesis step of nucleotide excision and base excision repair and has been implicated in recombinational processes in the cell. Thank you, sir. This one, sir. Uh, for elongation, in a base excision repair, delta in lagging strand it is used. Leading epsilon. Hmm. I think there are multiple replicons per chromosome. Thank you, sir.
So she is not there. Uh, no, no, sir. She is not in the home actually. Okay, sir. Sir, is it visible? Good evening, everyone. Myself, Qureshi Farhanaz, and my roll number is 24. Uh, and my topic is assembly of newly replicated DNA. So let's start with small intro about DNA replication. It is a very important biological process where one DNA can replicate into two new DNA. DNA replication is important for growth and repair of organism. Uh, DNA replication was postulated by Watson and Crick and they proposed the DNA double helix model. But with the help of Messelser and Stahl experiment, we got to know that DNA follows semi-conservative method of replication, where the DNA strand is separated and each act as a template for the synthesis of new complementary DNA strand. So, assembly of newly replicated DNA is done in eukaryotic cells into the nucleosome. What is nucleosome? Nucleosome is a structural unit of a eukaryotic chromosome consisting of a length of DNA called around a core of histones. Basically, a nucleosome is an um, is a section of DNA that is wrapped around a core of proteins. Inside the nucleus, DNA form a complex with protein called chromatin. So, what is chromatin? Chromatin is a complex of DNA and proteins that forms a chromosomes within the nucleus of eukaryotic cell. Uh, uh, How do nucleosomes assemble during replication? During S phase, replicated DNA must be assembled into the nucleosome using both newly synthesized and parental histone in the process tightly coupled to DNA replication. This DNA replication coupled process is regulated by multitude of histone chaperones as well as histone modifying enzymes. So, what are histones? Histones are highly basic proteins abundant in histidine and lysine and arginine residues that are found in eukaryotic cell nuclei. There are five types of histones named H2A. H2A is a histone important for packaging DNA into chromatin. H2B is a H2B histone is a structural protein that helps organize eukaryotic DNA. H3 histones plays an important role in activating the spindle assembly responsible to defect in mitosis. H4 H4 histone is also an important protein in the structure and function of chromatin. And lastly, H1 histone is also there as a linker histone which binds the entry and exit sites of DNA on the surface of nucleosome core particle and completes the nucleosome, and also it maintains the overall chromatin structure. So DNA packaging, histones are protein that are responsible for DNA packaging. The DNA wraps around the histones. Histones are positively charged proteins, and hence they can easily bind with a negatively charged DNA. So they form into two types. Histone tetramer. The tetramer is the central protein component of the core nucleosome. Histone H3 and H4 are the core components of nucleosomes. Histone octamer is the 8 protein complex found at the center of nucleosome core particle. These nucleosomes are wrapped together in a spiral structure called solenoid. Solenoid is a structure where the nucleosomes fold up and are stacked, forming a helix-like structure. So, uh, assembly of newly replicated DNA into nucleosome is the process by which newly synthesized DNA is assembled into nucleosomes immediately following DNA replication using both parental and newly synthesized histones. When eukaryotic DNA is replicated, it complex with histone. This requires the synthesis of histone proteins and the assembly of the new nucleosome. Transcription of new histone genes is initiated near the end of G1 phase, and the translation of histone protein occur throughout the S phase. And H3 and H4 tetramer is reused in one new strand, where H2A and H2B is broken down into two dimers, which are reused automatically. Thank you, sir. In which? Yes, sir. जीवन phase में end of the जीवन phase. Yes, sir. No, sir. They have histone-like proteins, histone-like structures, which have amino acids lysine and arginine. Thank
Thank you, sir.
Thank you, sir.